So yes, very good morning one and all. So today we are going to discuss again on the topic genetics. This is genetics 2 bridge course. In this session we are going to learn on dihybrid cross, its applications, exceptions of the third principle given by Mendel, and we are going to exercise the numerical types of questions, application based of questions of genetics along with previous year questions. So yes, if we have to begin with the dihybrid cross, once let us recall the yesterday's class as well, wherein we have discussed about monohybrid cross, right. Tell me what is the genotypic and phenotypic ratio of monohybrid cross? Very good. Phenotypic ratio is 3:1, genotypic ratio is 1:2:1 is the genotypic ratio. Fine. So yes, you got both phenotypic and genotypic ratio, right beta. Now if you have to see what are the laws that Mendel has given based on the monohybrid cross. Two laws he has given, right. What are they beta? First one is Mendel's law of dominance, second is Mendel's law of segregation. Okay. Second is what? Mendel's law of segregation, and first is Mendel's law of dominance. From Mendel's law of dominance, what did Mendel say from Mendel's law of dominance? That characters are controlled by discrete unit called as factors. Okay, characters are controlled by discrete units called as factors, and factors occur in pairs. Of these pairs, one is dominant over the other. Dominant one is called as dominant allele, recessive is recessive allele. This is given in the dominance, law of dominance. Law of segregation, of two pairs of factor, okay, factor exists in pairs. From that pair, each allele will segregate independent to each other, and there is no blending in the alleles. In the pair of factors, there is no blending of alleles. That is law of segregation. Of these two, which is universal? Law of segregation is universal. Right now beta? Now, these are the laws given by Mendel from monohybrid cross. Tell me what are the exceptions of monohybrid cross? Very good. First we will consider incomplete dominance. Okay, Nama? Incomplete dominance. Second is what? Online students also keep answering beta. What are the exceptions of Mendelian monohybrid cross? First is incomplete dominance, second is codominance, and very good, multiple allelism, and very good, pleiotropy. Be louder. Multiple allelism, and fourth will be pleiotropy. Fine, ma? So yes, in incomplete dominance what is happening? Dominant allele is not completely masking the effect of recessive allele. What is the best example of incomplete dominance in our NCERT? Flower color of snapdragon or antirrhinum. Examples are very important, okay, and those examples which are kept in the NCERT explaining the concept is again important, okay. Flower color of snapdragon or antirrhinum plant. What is the genotypic and phenotypic ratio here? 1:2:1. Codominance example what we have learnt beta? ABO blood grouping. In that, which is showing codominance? IA, IB combination of blood group, okay, wherein both of them are dominant to each other. Next. So, from these two, amongst these two, what is the genotypic ratio and phenotypic ratio? 1:2:1. Okay. Now, multiple allelism. Okay. What does multiple allelism concept mean? Multiple allelism, where one gene is controlled by more than one alleles. According to Mendel, what does he say? Characters controlled by discrete unit called factors. One factor is having one pair, okay. Two contrasting alleles will be present, or alternative forms also you will say, right. But In multiple allelism, what do you see? One factor is having more than one allele, okay, or more than one allelic expression. That is multiple allelism. Best examples we have learnt is starch synthesis, okay, starch grain size and starch synthesis. Second example is what type of disease we have learnt? Phenylketonuria, which is an autosomal recessive disease. Do remember that, okay, autosomal, loud, autosomal recessive disease. Clear, right? Next you tell me what is pleiotropy. According to him, one gene is showing one effect, one gene, one effect, one gene, one enzyme is the concept. But pleiotropy says that one gene showing multiple effects or one gene showing many effects is called as what? Pleiotropy. Okay. So, here one gene, many effects. Clear, beta? Best examples for pleiotropy, sorry, phenylketonuria is the example for pleiotropy, I am sorry, okay, wherein one gene is showing multiple effects. Under multiple allelism, the example we have learnt is again ABO blood grouping only, okay, wherein this is having more than one alleles, right, IA, IB and IO. So, for codominance and multiple allelism, what is the example, beta? ABO blood grouping. For pleiotropy, it is phenylketonuria and starch synthesis. Okay. All the examples, phenotypic, genotypic ratios should be in your mind. Fine. Now, let us explore dihybrid cross. Fine. Dihybrid, the name itself is suggesting that how many characters he is considered? Two characters or two traits are considered. What are they? Seed color and seed shape. Seed color and seed shape. Seed shape can be round or wrinkled. Seed color can be yellow or green. Fine. So, this will be the traits, these are the traits which he has considered. So, now how you have to look into is, we all know that parental generation is purely homozygous, yes or no? It is purely homozygous. So, these are the gametes from the parent, dominant and recessive, homozygous. When they are crossed in the F1 generation, this is the condition. Okay. What is that? Capital R crossed with self-pollination, right? Now, after this cross, one question, okay. How many zygotes are possible for this cross? First question, okay. Second, what are the types of gametes present here? How many types of gametes are there here? Okay. So, here two questions, beta. Question number 1, question number 2. First question is what? How many types of gametes are there? Types of gametes here in this, okay. How many types of gametes are there? Second is how many offsprings are formed or how many zygotes are formed with this cross. This is the question. How you have to identify, I will tell you, see. So, here if you have to see, look into it carefully, ma. The first question can be answered like this, types of gametes. Let us consider one parent, okay, one parent we are considering. This one parent, how many types of gametes, though there is a formula 2 to the power of n. What is the formula? 2 to the power of n, where n is the number of heterozygous condition. Okay. How many heterozygotes are there here? 2. 2 to the power of 2. Same you do for this parent also. Here how many gametes are possible? 4. For this parent also how many gametes are possible? 4. Now you got it. How you will identify types of gametes? By the formula 2 to the power of n. Okay. So, here for both the parents you have applied the formula, that is 2 to the power of n, to identify the types of gametes, and you got for male parent it is what? 4 gametes. For female parent also how many? 4 gametes. Now, we got to know how many types of gametes are undergoing fertilization. Okay. In F2 generation, how many zygotes will be formed? That is the question, right? This has a formula that is gamete square. What is it, Nana? Gamete square. Okay. So here female parent has how many gametes? You tell me. 4. Male parent has how many gametes? 4. Okay. So 4 plus 4, or you can consider 4 into 4. Or you can say 4 4s are how much? 16. Gamete square. You are multiplying the number of gametes. So total you will get 4 square is how much? 16. Fine. 4 square. Gamete square will give you number of zygotes. How many gametes are there? 4 gametes. 4 square is giving you number of zygotes. 4 male gametes, 4 female gametes. Square it, you will get number of zygotes. Have you got these two formulae? There are many formulae, but as this is the basic class, I am giving you the basic idea on dihybrid cross. I think this sort of idea you will not get with your IPE. Okay? But whenever you are going through dihybrid cross, use this formula and then you look into, then only you will understand why here you got 4 types of gametes, how you got 16 zygotes over here. Okay? Online students, is that clear, beta? So now you see, first in the parental generation it is homozygous, clear na beta? In the F1 generation you got heterozygous condition, which is resembling what phenotype? Dominant phenotype, clear? Now here in the F1 generation we will do self pollination. In self pollination, to do, first we have to know how many types of gametes are there in both male reproductive structure and female reproductive structure. Types of gametes, if you have to consider, 2 to the power of n is the formula, you got 4 and 4 types of gametes. In F2 generation, how many zygotes? The formula is what? Gamete square, 4 square if you do 16 zygotes. Now from those 16 zygotes, we have to see what is the phenotypic ratio and genotypic ratio. Just by looking into this Punnett square is given by whom? Reginald E. Punnett. Okay? So just by looking into Punnett square, you can say that there are 9 round and yellow, 3 round and wrinkled, round and green, and 3 wrinkled and yellow, and 1 wrinkled and green. This will be the set of combination. Okay? But from this 9, what is the generic combination? This I will tell you in your objective perspective. Okay? So from this 9, if you have to take 1 is homozygous dominant, and here also you will get 1 homozygous recessive. Okay? And here 4 are heterozygous dominant. Okay. 2 are 1 homozygous condition and 1 heterozygous condition. Again rest 2, 1 heterozygous condition, 1 homozygous condition. Understood? Now here if you have to consider this 3, what is the genetic combination? I will tell you. 1 will be homozygous only, small y, small y. 2 will be heterozygous 1, homozygous recessive other. Okay? This if you have to consider. Okay? 1 will be homozygous only, 2 will be homozygous recessive and heterozygous dominant condition. Have you observed this? Understood, right, beta? So now if you have to see the phenotypic ratio, it is 9:3:3:1. 9 phenotypically it is round and yellow, 3 are round and green, 3 are wrinkled and green, wrinkled and yellow, very good. 1 is wrinkled and green. Okay? Of this generic ratio is very important from Punnett square, because based on this only you will get the questions. Okay? How you have to calculate, I will tell you. But this trick should be in your mind. Okay? Wherein if you see from 9, how much, one thing you keep it in your mind, ma. 1 is homozygous dominant, 1 is homozygous recessive. These 2 things you keep in your mind. 4 will be heterozygous condition. See all these 4 from here to here, these are heterozygous dominant. Okay? 4 are heterozygous dominant. No? Of 2 are homozygous 1, homozygous other. capital Y, capital Y. This condition 2 will be there. Fine. Understood the generic combination of this 9 from this 3 and 3. One is completely homozygous, dominant heterozygous, recessive homozygous, dominant heterozygous, recessive homozygous. Understood now beta? Clear, right? Yes. Now this is all about the dihybrid cross. Tricks to remember the all the generic combinations from the Punnett square. Now there are some exceptions for dihybrid cross also. Like how there is there are 4 exceptions from monohybrid cross. Let's recall them once again. One is incomplete dominance, codominance, multiple allelism and pleiotropy. As such we have 4, right? Now here from dihybrid cross Mendel has given law of independent assortment. What law beta? Law of independent assortment. Law of independent assortment. What does law of independent assortment mean? Okay, when during the time of segregation or when the genes are passing from one generation to the other generation, the movement of the alleles is independent of each other, okay, at the time of gamete formation. When gametes are being formed from the parents, like R is it always moving with capital Y? No. Round is always with yellow? No. Round is sometimes with green also. Wrinkled is with yellow, wrinkled and green is also there. So that movement of gametes at the time of gamete formation, allelic combination at the time of gamete formation is independent to each other, okay. That is said in law of independent assortment by Mendel from what? Dihybrid cross. As such he has given 3 principles. But polygenic inheritance is again an exception for Mendel's law of inheritance. Okay, how is it an exception? According to Mendel, one gene is having 2 alternative forms. Yes or no? One gene is having 2 alternative forms. And one gene is coding for one character or trait, like that. According to pea plant, because it has one tall and short, that is what Mendel has considered. But if you have to take few characters or traits like human height, how many heights are there, like beginning from 3 or 4 to 7, 7.2, like that also tall or tallest people are there. So you can see variable amount of heights, variable amount of skin textures. So these many variants are possible by many genes, okay. Many genes coding for one trait is called as what? Polygenic inheritance. The name itself is saying no beta, poly means what? Many. Genes coding for what? One trait or character. Let us suppose height, it is coded by many genes. Let us suppose skin color, coded by many genes, okay. Same like in plants, if you have to take kernel color in wheat, coded by many genes as such. So traits are controlled by 2 or more genes, that is called as what? Polygenic traits. Don't get confused with these 2 terms, polygenic inheritance and pleiotropy. With these 2 terms you don't get confused. Pleiotropy means one gene showing many effects. Polygenic inheritance, many genes coding for one trait or character. Clear now beta? So inheritance of this polygenic traits, you will call it as what? Polygenic inheritance, or you can also call it as quantitative inheritance. And the genes are called as polygenes. Okay, genes are called as polygenes. Dominant alleles will show additive or cumulative effect. Okay, so here as one gene is giving many effects, there are many alleles as well. Of them, dominant alleles will show combined effect. Okay, wherein for example, if we have to take skin color, I'll say you in skin color, if this is the genic combination, okay, this gives complete black color or negroes, you can call it as. This combination gives you the skin color of negroes, okay, whereas this combination will give you complete mulattoes, means completely fair people, okay. But then the combination of dominant alleles, if this is the condition, they will be intermediate. But if this is the condition, they will be more dark. So, here if you see, dominant alleles are showing cumulative or combined effect. Understood ma? Clear, right? So, this is polygenic inheritance. Those traits are called as polygenic traits which are inherited, and this is polygenic inheritance, also called as quantitative inheritance, because dominant alleles are showing combined effect or cumulative effect. Best examples, and the genes which are passed on are called as what beta? Polygenes. Example include skin color in humans, height in humans, kernel color in wheat. These are the examples. Fine. So, this is about polygenic inheritance. Once let us recall quickly once again Mendel's 3 laws of inheritance. Okay. From monohybrid cross he has given law of dominance and law of segregation. This is universal. Okay. This many times why I am repeating though, this is very basic. But if you are not understanding this na, complete genetics will be a question mark to you, that is why. From dihybrid cross he has given law of independent assortment. Deviations or exceptions you have to consider. What are they? Incomplete dominance, codominance, multiple allelism, pleiotropy. Incomplete dominance example you tell me, beginning of the class we have learnt, no? Yes, snapdragon, or we can also call it as what? Antirrhinum, snapdragon or antirrhinum. Codominance, ABO blood grouping. In that what nana? IA and IB na beta. Next, multiple allelism if you have to see, ABO blood grouping only. Very good. Pleiotropy, phenylketonuria, which is autosomal recessive disorder, and starch synthesis. Fine. These are the examples. Polygenic inheritance we have learnt 3 examples now. What are they? Kernel color, skin color and height. Clear na? Fine, right? Now, so this ratios you have to remember. Okay. What are the ratios over here? First, type of cross. In monohybrid cross, what is the phenotypic ratio? Genotypic. When you are unable to remember dihybrid, trihybrid cross, you can use one trick, that is to just square 3:1. Okay, 3:1 into 3:1 will give you 9:3:3:1. Just square 1:2:1, you will get this. Fine. For trihybrid cross, if the question comes, just cube this. You will get the phenotypic and genotypic ratio of dihybrid cross. That is a small trick. Okay. Now, test cross if we have to consider, test cross is to define what? To identify the genotype of the individual. Genotype of what generation beta? Specifically F1 generation, or you can also say to identify the genotype of dominant phenotypic organism or F1 generation, whether it is homozygous dominant or heterozygous dominant. The genotypic and phenotypic ratio will be same here, 1:1. Incomplete dominance and codominance also it is 1:2:1. For polygenic inheritance, this will be the ratio, and this ratio will give a bell shaped curve, okay, where 1, 6, 15, 20, 15, 6, 1. So, this gives you what? From complete homozygous dominant to homozygous recessive, all the allelic combinations. In that allelic combinations, you will understand that all the dominant alleles are showing cumulative effect. Fine. And in this session, we have analyzed 2 important formulae. What are they? First is to identify the types of gametes. What formula we are using ma? 2 to the power of n, where n is the number of heterozygotes. Next, second formula we have used is? Yes, second is gamete square. Fine. So, this gives you what? Number of zygotes. Clear, right? Understood, no? Now, questions. Answer this, very important and very useful questions. Syed Rehan. Yes, good afternoon. Apart from ma'am, you say anything? Yeah, what's the doubt? Heterozygous round yellow seeded pea plants. Heterozygous round and yellow to you also do. Heterozygous round and yellow, what will be the combination? Fine, need not to worry, okay. First you analyze and think it properly, you will get it, okay. So, heterozygous round and yellow seeded pea plants were selfed, okay, they are selfed. This will be the combination, right, and they got total how many seeds? 800 seeds according to the question, okay. What is the total number of seeds with first dominant and second recessive? First dominant and second recessive, only phenotype he is saying, okay. First dominant means it can be like this or it can be like that. How many are there for 16 zygotes? First dominant and second recessive. I will show you. This. Look into it properly, no problem. Analyze and answer. First dominant and second recessive means this, right? First dominant, second recessive. This is also first dominant, second recessive. As such, how many we got beta? 3. Okay. For how many? 16 zygotes or 16 seeds you can consider. For 16 seeds, such combination is how much? 3. Same cross, he is asking you for how many seeds? 800 seeds. Okay. So, when it is 3 such combinations are there, okay, it is 3 for 16 seeds, for 800 seeds, it is how much? Okay. It is very simple, 800 into 3 by 60. Okay. Just tell me, calculate and tell me. $150, right? Understood or not how you have to calculate? This is just one sample question. So, here the answer will be 150, okay. Same thing ma, you have, that is why I said you, why this is important to know the allelic combinations, the generic combinations of this phenotype. Why is it important to know? Because of this reason only. If you know the generic combination for 16 seeds, for how many ever seeds he is asking also, you can apply and you can answer, okay. So, if it is for 800 seeds, now it is easy to calculate, okay. Now, second question, understood? No. Next, what will be the possible blood group in children from parents with B and AB blood group? With B and AB blood group. To see the maximum combinations, what we have to do is, let us suppose for male we will take B blood group only, no? For female, AB means definitely it will be this combination, right. So, now you tell me what will be the answer. Online students, you can also answer beta. So, here you can see the what will be the combination, say, do and say beta. So, it is easy for you if you can draw a checkerboard if possible. So, for male what are we taking? IB and IO, right. For female, IA and IB. Tell me the combinations. Tell me what blood groups you got? AB, B, A and B, okay. So, you got AB, B blood group, A blood group and B. So, you got A, B and AB blood group. What will be the option? You have understood or not? Okay. Just giving the blood groups to explore maximum dimensions, you have to take heterozygous only, because he did not mention it as like B blood group homozygous condition. So, you can take it like that and just cross and see, you will get. If you are unable to do this cross initially, take the help of Punnett square or checkerboard and you can do. Okay. Next, see this. Phenylketonuria is an inborn error of metabolism. Very good. And when we are dealing with genetic disorders, autosomal, allosomal, we will deal it more clearly. This you see. What is the pattern of inheritance for polygenic inheritance? Polygenic trait. Is it Mendelian? No. Is it X-linked recessive? No. Is it autosomal? No. Then what will be the answer? Non-Mendelian inheritance pattern. This you see. Asked in 2025. This also asked in 2025. See, when such questions are given, if you look all the options all of a sudden, it will be confused to you. So, just eliminate the unnecessary options, then it will be easy for you to identify the proper option. Next. Online you also answer beta. Genes R and Y follow independent assortment if capital R, capital R, capital Y produce round and yellow seeds, and this homozygous recessive are producing wrinkled and green. This is explaining what? Parental generation of which cross? Dihybrid cross. Okay. What will be the phenotypic ratio of F2 generation, he is asking. Phenotypic ratio of F2 generation will be what? That's it. Okay. This you see. This is from our basic thing. Take 2 minutes time and answer. There is no hurry. Take 2 minutes time. Bioverse A. Very good. Try to answer this question, ma. NEET 2024 question. Just from the basics of the chapter. Which one of the following can be explained on the basis of Mendel's law of dominance? Okay, he is only asking you from Mendel's law of dominance. Answer that. First, out of one pair, we have to pick out the correct statements, okay, which is explaining or which is supporting law of dominance only we have to take, okay. So, first if you have to take, out of one pair of factors, one is dominant and one is recessive. Yes, no, he said that. Second, you see, alleles do not show any expression. Is that true? No, alleles show expression, two contrasting expressions. So, this is wrong. Next, factors occur in pairs, true, right, in a normal dihybrid plants. The discrete unit controlling a particular character is called as what? Factor. Correct only, no? Next, the expression of only one of the parental characters is found in the monohybrid cross. Correct, right? Excuse. So, now if you have to see, pick out the correct options, it is A, C, D and E. Fine, easy. So, whenever that statement based questions come, just apply and see, look into each statement properly, then you can analyze, ma. Now, again blood grouping, this time you have to do and say me. Same type of question. So, ABO blood grouping he said beta. For father he is saying B positive, mother A positive, child is O positive. The respective genotype can be for father, mother and child. When father is B and mother is A, child is O, then both should be IB, IO, IA, IO condition, right? Do it and say father, okay, mother, look into the offsprings. So you got IO blood group in the offspring. So this you can confirm. This genotype you can confirm. Okay. What will be the right answer? A. B, can you take homozygous condition? No. Very good, Rehan. Next, AB, IA and IB is not possible, right? This is also not possible. This is also not possible. So what will be the right option? Next. This you see, statement based. NEET 2022. Last question. Mendel studied 7 pairs of contrasting traits in pea plant and proposed the laws of inheritance. Correct, right? 7 characters examined by Mendel in the experiment on pea plants were seed shape, seed color, flower color, pod shape, pod color and flower position, stem height. Correct only, right? All the 7 traits has how many varieties? 14 varieties. So here both the statements are correct. Option 1. Okay. So now you have got the basics of genetics clear, right? Both dihybrid, monohybrid, exceptions of Mendel's laws. We have exercised only 2 formulae, but there is so much of concept in that to learn more. We will do it in our advanced sessions. Okay? Yes, thank you. Thank you.